Thank you all so much for being here this afternoon, and thank you, Chris, for that kind introduction. My name is Nupur Rajay. I direct the multiple myeloma program at Mass General, and I'm super excited to have a fantastic panel who will momentarily introduce themselves. And I'm also excited to have Alex Stranahan from Bank of America, who's going to be a co-host for this panel discussion. And before we get into the panel, I just wanted to kind of highlight how excited we are about cell therapy. You know, the fact that we're using cells as a modality of drug treatment, live cells, which we are modifying either to create CAR T cells or genetically modifying them and then using them to treat disease is really pretty amazing. But I also do think that we are so much at the, we are barely scratching the surface. And what we're going to do this afternoon is really dig deep and talk about some of the gaps, some of the future things that we have in mind. So without further ado, I'm going to hand it over to Alec, who's going to start with the first sort of introduction into this panel discussion.
Great. Thanks, Nupur. My name is Alex Stranahan. I'm a senior biotech analyst at Bank of America, covering SMID caps. And I'm pleased to be joined by our esteemed panel of experts, many of whom are actually leading the charge for the next generation of gene editing and cell therapies. So the topic of this session is the future of cell therapies for liquid tumors. And until now, we've got 6 commercially approved autologous CD19 and BCMA-directed CAR therapies for lymphoma and multiple myeloma. But I think the consensus in the field, and I'm sure many of you would agree in the audience, is that we're just sort of scratching the surface of what this therapeutic modality could potentially do. But it's not without its shortcomings, right? So it's highly efficacious, functional cures for some difficult to treat patients, but there's also safety issues that it carries with it. And we're going to unpack all of this as well. And there's also the cost and the time, right? So there's alternative immune stimulatory approaches like bispecifics from a commercial perspective that could be competitive. And until now, it's, these therapies have been reserved for the most advanced patients. Although, I will say at ASCO, we saw the ZUMA-7 update for Gilead's CAR T in the second line, and that's a doubling of PFS, right, and a significant improvement in OS as well. So maybe just to start, we can jump into the current data, sort of where we stand with the current therapies. And maybe going first to you, Chip, and we can go down the line. And when you answer the question, please just give like a brief introduction as well. But looking at the latest data coming out of the autologous CAR T-cell therapies from ACR and most recently ASCO, I think they really help set the stage for how far the modality has come and where we are today. So, Chip, maybe what are your impressions of the data so far?
Yeah, thanks, Alex. So just brief introduction, Chip Baird, Chief Financial Officer for 2seventy bio. We spun 2seventy bio out of Bluebird Bio about 1.5 years ago to focus squarely on autologous cell therapy. We have a broad pipeline. Our most advanced product is Abecma, which is one of the 6 or 7 approved products in the cell therapy space. It's the first-in-class therapy for targeting BCMA and multiple myeloma. We've been on the market for about 2 years. The impressions from ASCO, I think 3 themes for us. Within myeloma, I think there continues to be a lot of enthusiasm for CAR T therapy. And as you think about BCMA and different approaches, whether it's a cell therapy or bispec, that if you have the product, and that's a big if, and we'll get into supply and demand later, it's preferable to sequence the CAR before other approaches. So there is sort of treatment and retreatment, and how that all layers, I think, is going to continue to play out. But certainly a lot of enthusiasm for the product, broadly speaking. We continue to have to work on the supply side, and a lot of energy moving towards earlier lines. Certainly the Gilead data supports that as well. In the CD19 space, we also have a play there that's in phase 1, and despite the strength of the data there and the flattening of some of those curves, there continues to be a lot of interest in new targets and new approaches, and so it's I think far from over in terms of having one treatment option there. And then within the solid tumors, we have a product moving towards ovarian cancer, and while challenging, I think there's tremendous interest from physicians, from patients, of getting after solid tumors with these products. So we're excited about that as well.
Very good. And Steve, maybe turning to you, if you look at the recent data, and obviously Sana's been a key component of this, what are some of the key lessons that you see being learned from the latest that we have from the cell therapies?
Sure. Well, first of all, thanks everybody for joining us. And Alec and Nupur, thank you for inviting me. So my name's Steve Haar. I'm at Sana. I previously at Juno, so kind of been playing around in this CAR T space for a while. And first of all, I think the people who have been working in this field, and so many of the scientists and investigators, it goes back 30 years, have just done something spectacular, right? And when you really look at where the data are going and what we have, I still think we're going to end up with lymphoma and leukemia, hopefully myeloma, being something like hepatitis C. And with hepatitis C, the vast majority of people today can be cured with a very short treatment. And there doesn't seem to be a good reason why we shouldn't be able to do the same thing with these hematologic malignancies. And to be very clear, we're not there yet, right? I mean, I think around 100,000 people a year in the US and Europe die from those three diseases, and something like 4,000 people got a CAR T cell last year, right? And of those who got a CAR T cell, probably half benefited. So I think first and foremost, what we as an industry need to do is deliver on the promise of what we've already made. And that's going to require us to figure out the challenges of scale. And those challenges of scale might be solvable with some of the things that we're doing at Sana, which are around allogeneic CAR T cells or in vivo delivery. We're doing both of them. And they might be solved by some of the real great investments that companies are making in making better autologous cells. And then we have to make them better because it isn't good enough to get 40% of patients. We need to be at 80 to 90. And I think the tools are there, and we really do understand now why patients don't benefit and why they recur. And I think we can do that. And then we have to take it other places, as Chip said. So if the data from ASCO were just tremendous to see more benefit as patients are treated earlier. There's a bigger impact that these CAR T cells can have, but only if we can make them. And so we have to solve that. And then I think what you see emerging is that these CAR T cells can be at least as disruptive in autoimmune disorders as they have been in blood cancers. And that will be that's not the focus of this presentation because we're talking about tumors, liquid tumors, but that will be something that I think is going to be a big area of focus for, I think, the cell therapy community over the next 5 to 7 years. And the data that came out of Germany, where you're seeing basically cures, functional cures at 2 years, off all medicines with completely normal immune systems, is really spectacular. And it's a wonderful opportunity to help people who suffer from 30, 40, 50 years of a lifelong disease. And so that's what, as I look at what's really coming about, something that could be, just spectacular for what we're doing.
Great. And Laura, maybe lastly, just on this topic, what was most exciting in terms of recent developments? Obviously, Intellia is a leader in gene editing. Anything out of ASCO or ACR that you would highlight as being most exciting from you?
Yeah, and again, thank you for the opportunity to be in this panel. And so I'm the CSO of Intellia Therapeutics, and thank you for saying that. Yes, we are leading the charge in CRISPR medicines. We have really impactful data for our in vivo gene editing candidates, and actually this weekend we released additional data on performance in vivo, including what potentially could be functional cures in hereditary angioedema. But maybe what's a bit underappreciated is that we also have a large effort, probably equal in size to what we have for in vivo on cell therapies. We pay attention obviously to what's happening out there, and ASCO and AACR are two key conferences where we understand where is the field moving. What can we learn from competitors, right? What can we learn from academics, and understand what are the challenges that remain to be fixed. And for us, one of the challenges is ensure that whether it's a T cell or NK cells, for immuno-oncology or immunity, which, by the way, we do it internally and through partnerships. For example, with ONK Therapeutics and Avencell for immuno-oncology. And Steve was just referring these amazing data in Germany. This is our partner, Kyverna Therapeutics, is pursuing for autoimmunity for lupus nephritis using CD19 CARs. But the goal is understanding how do you need to endow your T cell or NK cells, right, with all the key genetic modifications that will make it, particularly for example in solid tumors. That I know it's not the topic today, but it's really important because that's a holy grail, right? How are you going to get to the tumor, overcome the suppressive environment, ensure that the CAR T is, or the TCR, right? They have on-target activity, low, off-tissue target activity, and you have durable responses. And for that, what's very important to us, there were two aspects of it. One is how can you cell engineer that, with the top quality, and for that we use CRISPR editing, both for insertion and for knocking out genes. And we have a proprietary cell editing that it's superior to what we have seen from others. And then use that to be able to engineer, for example, an allogeneic chassis, right? To which you could use for multiple applications, again, immuno-oncology or immunity. And there is continuously differentiating and testing what you have versus what's best out there, right? And the idea is to learn and potentially to leapfrog, because what the patients really need are efficacious, safe, and durable responses. And what we're seeing today is not there yet.
So, that is fantastic, Laura. We've all been really excited coming back from ASCO and AACR. Obviously made a lot of progress in the hematologic malignancy space. I love that you said, Steve, we're going to be looking at this as short-term treatment, and we're going to be curing a big subset of patients. All three of you are leading companies with very novel technologies, and I'm just wondering if each of you you've already done a little bit of this, but if you can speak to how you're going to use these novel technologies and see and address some of the gaps that you're seeing with gene and cell therapy as we have it today. And I'll start off with you, maybe Chip.
Sure. I think what's so exciting about the space that we get to work in, and as we think about the future of cell therapy development, it's a naked CAR is not going to be enough. And so, we're looking at potency enhancements, signaling enhancements, dual targeting, just to hit on one, a gene edit to create a CBL-B edit in our DLBCL program, and that's basically takes the brakes off the car and may confer a longer half-life and to the cell. So, those are the kind of things that, if it's true and it works in that subset of patients, that's great for that program. But then that's a tool that we can apply in other settings and in other indications. So, again, I think the ability to work on a variety of tools, both ones that we develop, ones that we in-license, I think is the future as we're thinking about tackling these ever more complex biology problems.
So along those same lines, Steve, your company, Sano, is doing a lot of work in the allogeneic space. And can you speak a little bit to how you're going to make those more accessible and more tolerable, given that it's an allo product?
Well, so our goal has been able to manufacture cells at scale that will function, graft, and persist, right? Ultimately across any cell type, very simple. And the most challenging aspect of all of that has been, I think, cellular persistence. And in particular, since the advent of transplant medicine, allogeneic rejection. And people have gotten around it in two different ways. One are autologous cells, which at the end of the day you can only use for cells that exist in suspension, more or less. And they're very, very difficult to scale. And we can get into like quality and all those things. Then the other side of it has been to just massively immunosuppress patients. And again, that is probably even more problematic from a scale perspective, just because of the risks of cancer and infectious diseases. So our goal was just to figure out how to evade the immune system. Pretty simple, right? And it turned out that people have been making a lot of progress on that. And we brought in a couple of different groups, and probably most importantly, a woman from UCSF, Sonia Schrepfer, who joined Sana at the outset. And I'm pretty comfortable saying that we've solved the problem of immune rejection for non-human primates, for mice and for humanized mice, and we can get into how. But we don't know if it really works in people. And we'll learn that over the next, actually, probably, I don't know, a few weeks, a few months. And if it is true, it becomes very, very clear how to, one, answer your question around scale. Because in every manufacturing batch, assuming that we think we have an idea what the dose will be, we make just around 500 patients worth of drug per batch, for cancer, and probably 1,000 patients per batch for autoimmune. So it's a very easy to scale. And it's not that different of a process than what an autologous CAR T cell is, right? And if that's true, then what we want to have, if we've evaded the immune system, is we should be able to use things like lymphodepletion that are the same or less than autologous cells, right? And get efficacy that is the same or better than autologous cells. Because in each of for an autologous cell, there still is problems of immune recognition of the CAR, right? And so that's the way we've gone about solving it. It's a little bit of hubris to think that we might be able to overcome the human immune system. And sometimes big bets pay off, and we'll figure that out over the next few weeks. And if it has, the beautiful thing is we can really quickly take that platform into a whole host of targets: CD19 for cancer, CD19 for autoimmune disease, BCMA, CD22, where we have all of those we have clinically validated CARs. We can do GPRC5D, where we've built our own, and hopefully it's what allows us to go into a whole host of other places. But it's really an immunology problem, and I think so often people have approached this from they have your original question, you have a technology or a tool, I can edit the genome, so I will, instead of really trying to solve the biology problem of how immune rejection happens, and then fixing the genome to grapple with that.
Yeah, I think you've highlighted one of the most important issues around allogeneic cell therapies. One is persistence, and you all are addressing how you're going to knock down some of the rejection genes. But that brings me to you, Laura. Your company is focused on gene editing strategies, and you've done that very nicely in disease models, and you've shown proof of concept there. How do you think you're going to move this forward, both in disease states as well as in some of the cell therapy approaches, using your CRISPR technology for us to be able to get rid of these, rejection signals, so to speak?
Yeah. So, we have assembled a toolbox of genome editing modalities, right, that go from traditional canonical cleavases, and that's a plural, to base editors that can serve a good purpose of multiplex knocking down genes, right? For example, in cell therapy engineering. And then we do have a DNA writer. But in addition to that is the ability of doing all the edits, not being restricted in how many insertions and how many knockouts you want to do in a cell therapy. And this is whether it's in vivo or ex vivo, but the topic today is ex vivo. So for that, very early on, there is a number of standard things in the field like lentivirus or electroporation that were not in scope for us, as we believe that CRISPR modification is precise and controllable, so you know exactly where you put it and how many copies of S do you have. And the other was electroporation. We find that electroporation by itself, it causes quite a bit of damage on the cells that you're trying to do. whether those are hematopoietic stem cells or T cells or NK cells. So we developed a non-viral delivery modality that we use, and we could do it in a multiplex once or in a sequential manner. So and all of these are scalable all the way through clinical ready scale material. So this is important because it gives you the freedom to really understand how you want to modify that cell. And for that, we combine different efforts that range from having in vivo immune-enhancing edit, in solid tumors, for example, right? Doing T cell libraries, CRISPR-I, CRISPR-A libraries to understand, what are the key genes that, you may need to knock out or knock in to endow the cell with, properties. The other is looking at the CAR Ts, right? So I think that there is first, second, third generation on the CAR designs, but there is a lot more innovation that can give in to look again for regulated important responses. And then there is another category, which are what are other things you may want to be expressing on the T cell, again, to lead to long-term persistence and particularly in a solid tumor, right? Overcoming the nutritional deficit, the anatomical barriers, the immunosuppression. So the ability to put all of those together is enabled by having this cell engineering platform under it. So that's the goal. And again, as I was saying before, is trying to, for example, on allogeneic, and I agree with Steve, right? I mean, we need to have better allogeneic therapies, right? So having something that lasts for a month or 2 months, I don't think that moves the needle, right? So you want to have something that's going to be persistent. Ideally, once and done, right? So that patients, whether it's 2 months or 6 months or a year after that, there is no recurrence. And as a field, I think that's where we need to go.
Great. And I want to switch gears a little bit and talk about manufacturing. This was alluded to before, and especially as we're thinking about cell therapies moving to earlier lines of therapy and potentially even to solid tumors, it does seem like manufacturing is going to become more and more of an issue. We're better than we were when CAR T was first commercially available, and I think a lot more centers have the capabilities to do an autologous CAR T infusion, but it still takes multiple weeks, right? So maybe Steve, actually, we can mix up the order. I'll start with you. As we sort of move earlier in the treatment paradigm or into solid tumors, what needs to happen from here for cell therapy to be successful, whether it's autologous or allogeneic?
Well, for solid tumors, I think you got a biology problem before you got a manufacturing problem, right? And the biology problem is that, very simply, we don't know what the problem is, right? Is we can't get the cells there. Do we not have the right targets? If we have the right targets, how do we get the cells activated? So we can kind of go through that. We have to solve the biology problem before we get to manufacturing. The manufacturing, it's having been around this place for a while, this field, it's challenging. And I think the first thing to recognize is that manufacturing is not really, it's not what we think about often. It's about buildings and things like that. It's a science, right now. And we have to improve our manufacturing science and our process characterization and product characterization in order for us to really effectively scale any of these things. And my outside view, and Chip will answer this better than I, around some of the challenges of autologous is it's not just about making more of them, it's challenges with the quality, which is a lot of people. If you could automate quality, so people think about ways to do that, you'd have a dramatic impact, because every single patient is their own batch record, right? And every single patient has to go through all of the release assays that you do for a huge 30,000 liter bioreactor for a biologic. And that's something that can be very time consuming and challenging for companies. All that being said, there's just been a ton of progress. My own bet for a long time was that it would be much easier to solve the complexity of supply chain than the complexity of biology. I think that over the last few years, we've made enough progress in understanding transplant rejection that I'm optimistic that we can overcome this by some of these challenges by understanding how to make allogeneic cells that last at least as long and work at least as well as autologous cells. But we have to prove that. And if we have, then the manufacturing becomes a lot more straightforward.
Chip, anything you want to add from the autologous side?
Yeah, I mean, we spend a ton of time on manufacturing. It's probably the number one question that we get from the outside looking in. So a couple thoughts there. What you have to generate those 15 or 30 patients of proof of concept data and the skills that takes is just very different from what you have to do the same thing on the commercial setting and scale to thousands of patients. And that crossing that, as we call it, the valley of reality, is just harder than it looks. And again, there's only a handful of companies that have done it so far. I would comment that yes, it takes huge capital investment, but it's a huge people investment too. And just to make a step up in terms of your capacity, the operators in the plant, you're hiring them 3-6 months out and training before they're even eligible to be on the floor in the commercial setting handling a patient's cells. And that work is intense. It's over a period of weeks, and one wrong step there, now we don't have a product that meets spec, and now we can't deliver for that patient. So manufacturing takes a ton of effort. That being said, we are figuring it out, and together we have a competitor in the BCMA space. We'll treat about 2,000 myeloma patients this year in the U.S. There's about 4,000 patients on the 5th-line plus label. So we're still not there. And that label in myeloma is going to expand most likely for us and for our competitor to something more like 16,000 patients on label by the end of the year. So we're going to be, even with 100% increases in capacity, years before we get to the moment where if you want a car, you can get a car. And that's the challenge that we're going to be facing for some time. The other thing, when you look at all the players that have been through this, in the clinical setting, you have one set of specs and release specs and what counts as a product. Almost every sponsor goes through in the commercial review, the regulatory review, those specs get tightened. And so out of the gate, it's a lot harder to deliver the same cells in the commercial setting. So again, everyone goes through their growing pain, but we're, I would say we're on the crest of that hill and starting to run downhill in terms of seeing those capacity expansions, which is really exciting to deliver for patients.
Laura, any advancements that you're seeing that's feeding into manufacturing, gene editing, or otherwise?
Well, maybe in manufacturing, what I was excited to see at ASCO, right, was, the short manufacturing, right, for autologous. T-charge. And again, although I'm developing allogeneics, I think it's really important that we have autologous therapies to patients today. And, some of the, the advances on really shortening the time, even to 2 days or something like that, right? Which also shows you that you don't, with a smaller dose, right, you could have efficacy. So that was good. The other is, how, companies are investing in, closed manufacturing, systems, right? Part of the idea is how are you going to have it access, right, by having it more distributed to the patient, where the patient is. I think that, again, ultimately, many of those issues are going to be overcome by having off-the-shelf therapies, right? Where you select, and these are allogeneic therapies at work. The benefits is that, those T cells, they come from healthy donors that you can pre-screen, so they will have, the proper properties, immune properties, to give you a good T cell therapy. So, this is, and also, of course, they have never been exposed to disease, right? And prior lines of therapy. So, those T cells are going to be in much better shape to do the job and to be expanded. And for a patient, right now, for some therapies, they need to wait, 30 days, 40 days, right, from vein to vein. And, in a significant percentage of the patients, either, the therapy cannot be manufactured, or by the time that you're ready, they progress. So, again, there is, technology evolution, right, and solutions that come with that. So I'm looking forward to a future where, you could be infused, 2 days later after diagnosis.
Yeah, and to speak to that, Laura, we saw a little bit of that at this year's ASCO, where we have the fast CAR. We also have the T-Charge CAR T-cell, where they're looking at in vivo expansion. We've talked a lot about allogeneic CARs, and all of you, 3 of you have been leaders in this field with your innovative platforms at your companies. And that then brings me to another sort of question. And I think we've been doing this, academic medicine, collaborating with all of you, which is where we've gotten to both gene therapy and cell therapy. So where do you see this future of collaboration, and how would you like to see us improve upon that? And I'll start off with you, maybe, Chip, because I've worked a lot with Bluebird Bio, and now 2seventy Bio. So how do you think academic medicine should be working with you guys to make these processes better?
Yeah, I mean, academic collaboration has been a cornerstone of my prior company, Bluebird, and the current company here at 2seventy. And I think from having KOLs in the field who are excited about the product, who are proselytizers, who can be on the front lines of convincing very sick patients to take a calculated bet on an experimental medicine like this that is non-traditional, that takes a lot of explaining. I think that was just so important in the early days of Blue, and even today with 2seventy. I think a lot of the collaborations we have now, one with Seattle Children's, they're able to do their own manufacturing, so that from a turnaround time, from asking and answering and iterating, I think a lot of what we do, the first patient to the third patient to the 10th patient, the product, the process evolves, and I think having that on site with a committed investigator is just makes all the difference in terms of recruitment, and in terms of learning, and in terms of getting to a product that actually delivers for patients. So, we're always looking for more there. The other thing is, as we're internalizing manufacturing, we're getting approached more by other academic investigators saying, hey, I have a product, I have an idea, we don't have an ability to make it, we can talk to a third-party CDMO and pay cost plus 40%, or we can do it with a committed partner who, can bring capital, can bring resources, and invest in the future. And so that's, it's going to be a cornerstone of our business going forward.
I think for the future. So just to speak a little bit more to that, Steve, discovery and then product development, where do you see that sort of balance out with academic medicine and industry?
Yeah, so first off, we're part of the same ecosystem, right? And I think we have some very aligned goals, really around trying to make disruptive medicines for patients. And I think often that facet is lost upon so many parts of society. And I think the more we can educate the world about where we've really made rapid progress is when academia, industry, and government have come together. And so figuring out how do we create those epic challenges where we can kind of align and do that. I think when I think about what you guys are best at in academia, I mean, you're the creative spark of all innovation, and so much around the way a great academic scientist thinks is you find a discovery, and you're always trying to widen that aperture to figure out, hey, how else can I apply it? Where else can I do it? And what we think about in industry is, if you have a discovery, is how do I narrow this down, the aperture, so there's 3 or 4 pages in a product label, so that you can use it when you're talking to your patients. And so I think we can solve different problems together by focusing on what we're really good at. And then we really need your help on the back end, because again, just those statistics around how many patients actually get these cell therapies or any disruptive drug it's disappointing, right? And you understand what the barriers are within your own hospital, but in particular, if you can help us get out in the community and ensure that we're accessing where most patients reside and where we can have the biggest impact. which is not just in the academic medical institutions, but really taking these disruptive therapies and getting them to where most patients live. And we need your help on that in a big way. So I think it's at the front and back end, and we can focus on the middle.
I'd be excited to collaborate with every one of you. Laura, did you want to add anything to that?
No, I fully agree with the comments made, right? And particularly focus on what you're good at, and then look for opportunities for synergy, right? So, and that synergy with academics as well as with other companies, right? So we cannot do everything. So, how 1 + 1 could be many, right? And ensure that you're, finding the best technology and the quickest path to value, not just, initial interesting data, but really to what's going to move the needle for patient care.
Great, and maybe the next topic, maybe just pulling this back to a higher level, and I honestly can't think of a better group of people to ask this question, but where do you see the next innovations coming from as it relates to cell therapies? Is it allogeneic, sort of breaking ground and really inflecting on the clinical data? Is it gene editing, really imparting novel and groundbreaking attributes to the cell therapies? Is it autologous really making a big commercial impact? Maybe starting with you, Laura.
Yeah.
You can come down the. Yeah.
So look, I do think allo, these, ready-made, high quality, that's going to be really impactful for the field. And not just for immuno-oncology. I think the opportunity on autoimmunity, and that's not just, B cells, for lupus, right? But, understanding how you make synthetic targeted Tregs that, that will make a difference. That's, and this is something we are working on. We're very excited about it. And the other is that how this opened the path to regenerative medicines, right? Where you don't need to have any, cartridge or anything to protect your, your cells, right? Where you could truly go and do, whether it's new organs or, new cells infusions or implantations that, will make a difference. So, the technology is moving towards that, and we are building on the knowledge, right, from different applications and expanding from this into multiple areas of medicine.
Steve?
So I'm going to answer your question with yes, with all those things. So maybe it depends on maybe what you think we're really good at. So if you said, well, what have we accomplished? I think we've accomplished the ability to gene modify T cells to go after liquid tumors. I think we've accomplished the ability to gene modify HSCs to fix genetic disorders. I think we've accomplished, again, this is an industry, the ability to deliver either DNA or gene modification reagents to the liver, right, to take care of genetic disorders. And those, if we put those in the rearview mirror and say, okay, those are done. Obviously, from an impact perspective, we're probably still in the first or second inning. But from a technology perspective, the field's figured it out. Then what really is coming next? I think, first off, if you can, the two biggest things we could figure out would be how to overcome immune rejection and make allogeneic cells. Because if you can scale T cells, and you can really now enter in the era of regenerative medicine, right? Because we've seen in the field that you can take stem cells and make them into cells that might be able to replace damaged or missing cells. But that really isn't scalable or effective unless we've overcome immune rejection. So if you've done that, you get both T cells and you get all kinds of other cells from stem cells. And I would say then the other is being able to deliver to other cells besides the liver. And I'm pretty confident that those two things will both be solved in the next, 2 to 3 years. And that the implications of that will be felt for decades. Just like the implications of the ability to insert a gene into a T cell. It didn't happen overnight, right? It's been probably 15 plus years of really slogging it out to make these into really beneficial therapies like what 270 is bringing forward.
Yeah, I'd answer the same way as Steve, which is yes. I think they're all, whether it's actually definitive wins in solid tumors or allo or in vivo gene editing, it's all coming. I'm not smart enough to say when that is, but it's the beauty of a panel like this. It's everyone's sort of on the team that way, and they're just different angles on innovation. I think it's just, if you were making bets, I think that, you know, autologous, where we spend more time today, is more of an engineering question of how do you scale. And so you can, like, can you scale? Yes. Can you do it more cost effective? Can you get to more places? All of that you can see, and you can kind of understand, like, what are the engineering steps you need to do to automate or reduce expansion time or improve the supply chain or whatever it is. You know, I think the question of evading the immune system, I think it's when, not if, but it's more of a biology question. That's harder to say when that moment's going to be, but there's enough people running at it that I also believe when, not if. And then I think, you know, we certainly are spending time on in vivo gene editing approaches, because the idea of sort of autologous in an in vivo kind of way makes a lot of sense to us, but how you deliver that vector, and you know, I think there's still work to be done there, but it's an exciting time. And you know, I think right now our belief is autologous cells work, and there's a lot of room, to Steve's point, we're still in the early innings, to make that even better on behalf of patients.
So, you know, speaking about T cells, we've been spending the last 35 minutes talking about T cells. There are other cell types in the body that we're all excited about, specifically NK cells, some of the other cell types. Does anybody want to take a stab at, if you're thinking about solid tumors, would you be thinking of a different kind of a T cell therapy or different type of a cell therapy? And the other thing, we've spent a lot of time talking about autologous and allogeneic, and from a clinician standpoint, I really think that they're going to be working in concert. Working with an allogeneic model as well as an autologous model is what we would like to see, because of the issues around persistence and rejection that we've talked about. So does anybody want to pick up on other cell types that we might be wanting to genetically modify?
Sure. I mean, we do have a partnership with ONK Therapeutics. So, you know, they're using some of our engineering to get all the modifications on the NKs. And, you know, and those are moving along. I think they're doing a great job. And, you know, and I want to have it all. So, you know, I'm excited about our T cells, but I'm also very excited about what our partners are doing. And, you know, the answer will be in the clinic, and I think there is going to be multiple answers. There is not a single solution for everybody. You know, the other thing is combination therapy, right? I mean, there is new data on, you know, on bispecific, but now on NK engagers, right? So, you know, it's up to you, the clinicians also, right, to help us understand, you know, what's the smart, you know, combinations and where the field needs to move to.
I know it's really hard for the oncologist to think about anything but cancer, but if I were to postulate where we'll see the next big impacts, it would likely be outside of oncology with cell types other than T cells and NK cells. And that you're going to see, you know, HSCs coming shortly, and then you'll see a number of stem cell derived products that if truly we have overcome immune rejection, you'll begin to see some level of efficacy from them. I think the real challenge that I've always had with both NK cells and solid tumors, NK cells, they work, but where do you use them, right? They don't really persist, and what do they do that's special? And what they do is special is they eat tumors, which is pretty good, but trying to figure out how to clinically put them in, I haven't yet figured out. And with solid tumors, I still don't know what the problem is, right? And until you understand, and it seems like there are probably 3 or 4 biology problems that we have to solve before we really kind of get there. And whenever, you know, if you just solve one, you don't even know if you've really solved it, because it still may not work, the drug. So I'd be really surprised if we can't figure out how to turn these T cells into something that will function against solid tumors. There's no reason they won't work. Checkpoint inhibitors work. We shouldn't give up. But the direct path to making a gene-modified, directed T cell work against solid tumors is something where I think we still have to grapple with, you know, 3 main challenges: getting them there, recognizing the tumor, and getting them to activate in a very toxic environment.
And any role of pharmacological manipulation, I think you guys are doing some of that with your cell therapy products? Chip?
Yeah, I mean, I just build in on what others have said here. I think in the solid tumor setting, our approach is with Regeneron as a partner is orthogonal, multiple products together. So whether it's a CAR with a checkpoint or a VEGF inhibitor, or I think, and again, I think we have to explore that in parallel versus sequentially. It'll be a whole nother set of panelists before we ever get there. So I think that we're interested in exploring in an orthogonal way as soon as we can.
Great. That was a fantastic discussion. Alec, do you have anything else to close up on?
No, well, I mean, I'm leaving more optimistic on the space, and I think it's going to be an exciting, next decade for cell therapy if all these things come to fruition. So thank you so much, guys, for participating, and thanks everyone for your attention.